we want to understand how cells control the different pHs in different parts of the cell. And it turns out that the main way that our cells control their internal pH is through an enzyme, a protein complex called the V-ATPase. The V-ATPase is effectively a proton pump. It uses energy to pump protons into different compartments in cells, controlling their pH. And if this process doesn't work well, a lot of what has to happen in a cell doesn't work properly. We use V-ATPases to pump protons in order to transmit chemical signals in the brain and the chemical synapse of the brain. We use V-ATPases to control the formation and growth of bones. We use V-ATPases to pump protons in order to control the function of the kidney. And if any of those systems aren't working properly, we get different diseases. We also want to be able to limit V-ATPase activity, because it turns out that cancer cells will often use V-ATPases in order to allow them to proliferate and invade and digest surrounding tissue. So we could understand what the V-ATPase looks like. We can design molecules to act as drugs, and those can be used to prevent cancer invasion and metastasis. So what we've done is we've isolated the V-ATPase from cells, we've purified it away from all other proteins, and put it into the electron microscope to study its structure, with the idea that if we can understand what it looks like, we can understand how it works. We know that this region of the molecule is the part that uses energy, chemical energy, and it converts that chemical energy into this rotation of this rotor. This portion of the molecule will sit in the membrane, and by spinning will pump protons across the membrane. Looking at this structure, we can start to understand how you might design a small molecule, a drug, in order to interfere with its activity. If you want to stop it from pumping protons, all you'd have to do is jam it so that it cannot spin. So this structure gives us insight into how we might come up with drugs to affect the V-ATPase activity.